Thank you for joining me. I always like to have an opportunity to share my research. And I'm going to be titling this talk An Ant Odyssey, which is going to involve taking a journey. In this talk, we're going to move across continents, from day into night, and into the underground realm. And the subjects of the talk are going to be confronting, as we all are, some important challenges that are related to extreme temperatures and global climate change. And we're also going to explore some challenges for the researchers that involve venom. And hopefully the subjects will bring us the promise of increasing our understanding of how animals are going to respond to global climate change. And I hope that you'll at least accept the possibility that sometimes to answer big questions, you need to go small. So my lab uses ants as research models in order to understand how animals are affected by climate and mainly temperature variation. So why do we do this? I'd like to talk a little bit about the power of ants as subjects for this research. One of the strong features of ants is their small body size. And because they're so small, ants are very sensitive to ambient temperature conditions. And they exhibit a wide range of behaviors and occupy different habitats. And ants are also social. And this is going to be important at the end of the talk when we explore how individual differences can impact the social group. So what does my lab do? One of the things that's important to the data that I'll be talking about today is we measure environmental temperatures. And we both record temperatures on the surface where the ants are active, and we can also place small temperature loggers that will record temperatures over longer periods of time at different places in the environment. And we also measure the thermal limits of the ants, and these data are going to be really important to this talk. What we're interested in doing is getting a handle on the maximum and minimum temperatures that the ants can tolerate. So we're going to refer to these as CT max, or maximum critical temperature, and CT min, or minimum critical temperature. So how do we measure these? Well, we place the ants in small vials, and then we heat or cool them in a controlled way. And what we're aiming at is getting a recording of the temperature limit at which the ant loses mobility, and we refer to that extreme temperature as the CT max or CT min for that individual ant. So we're going to explore patterns of environmental variation that are associated with variation in the ant's abilities to tolerate different temperatures. So in chapter 1, I'd like to explore the importance of moving into the subterranean world, and we're going to use army ants as subjects for this part of the talk. For us, the subterranean world is a place of mystery and oftentimes invokes fear, but we can also think, as any hobbit might tell you, the underground world as being a place that can be very comfortable and actually cozy. And that's the aspect of the underground world that we're going to think about in this talk. So the underground world buffers temperatures. Species that live underground are going to experience a more moderate temperature environment than species that live above the ground. And below ground species are going to experience both more moderate high and low temperatures. So here's some data that we've collected that illustrate these points. And the way to read this graph, we're looking across on the x-axis across the course of a day, and we're going to look at the ranges and the average temperatures that were collected by data loggers that were placed on the soil surface. So those are the red parts of the graph, and about 4 inches underground, that's the blue parts of the graph. And what you'll notice is, particularly in the middle of the afternoon, the maximum temperatures above ground are warmer, and the minimum temperatures in the evening below above ground are cooler, but the below ground environment is less variable at all times of day and night. And it turns out that army ants are good subjects for this research because different species vary in how much of their time they spend above versus below ground. And so this video clip is of a swarm raid of a surface active army ant, and you can see that these ants are, as they're sweeping across the floor of the forest, they're exposed to extreme temperature conditions. In contrast, this is a subterranean species, Labidocecus. These are very rarely seen above ground, and so here they've briefly come above ground, and you can see that they're frantically working to cover themselves with a little tube of soil. So these underground ants are usually sheltered from temperature conditions. So our predictions are that because the above ground species are exposed to and need to be able to deal with extreme high temperatures and extreme low temperatures, we expect them to evolve greater tolerance of all temperature extremes. So the patterns we expect are, if we look at a range of species that go from subterranean to above ground, the above ground species should have higher CT max, they should tolerate higher maximum temperatures, and they should also tolerate lower minimum temperatures. So we measured average CT max and CT min for 15 different species of army ants, and these range from fully subterranean species to fully surface active species. And the results are that the predictions were met. So for the above ground species, they had both higher CT max and lower CT min as expected. And the implications of this study are that below ground species may be more vulnerable to warming climates. And while we were collecting the data for this project, I learned an important lesson that army ants are not alone in the forest. So while I was out with students on a field course studying army ants, I was bitten from behind by a viper at a field station through what might nicely be called a series of unfortunate events. I wasn't treated for this bite for over 12 hours and was in pretty dire medical condition by the time I was given 10 vials of antivenom intravenously that probably saved my life. And you can read about this episode in the February 2016 issue of Natural History magazine. Okay, so for the second chapter, I'd like to talk about journeying from day into night. And we're going to be looking at how climates and temperature variation in the tropics are very different than they are in the temperate zone. And the important point here is that seasonal temperature differences are minimal in the tropics. So if we look at average daytime high temperatures, the difference between the warmest and the coolest month of the year is only about 2 degrees Fahrenheit. If we look at the average nighttime low temperatures, the differences are even smaller. The warmest and coolest month only differ by about 1 degree Fahrenheit over the course of the entire year. So this leaves us with a much larger gradient, a much larger change between day and night. So daily temperature differences in the tropics are much greater than seasonal temperature differences. So they're about 13 to 14 times higher than the seasonal differences. And so for animals, the difference between day and night that happens pretty much every day throughout their entire existence is much more important than seasonal variation. So what this suggests is that daytime differences might be very important to tropical animals. So we were interested in testing whether tropical ants' thermal tolerances differ over the course of the day. And we're going to focus in on the species in this photo here. This is the thief ant. And we observed that thief ant workers leave their nests to collect food at all hours of the day or night. And that means that they're exposed to both extreme high and low temperatures. So we collected subjects during the coolest and warmest parts of the day, and then we measured their CT max. And what we found was that consistently, daytime CT max was significantly higher for these ants than nighttime CT max, and the difference was about 2 degrees Fahrenheit. And we also marked and followed some ants from the same colonies and found that nearly all of them were active foraging both day and night. And what this suggests is that these CT max differences that we've observed over the course of the day are actually represent changes in the individual's ability to accommodate high temperatures over the course of the day. So it suggests that there's a strong individual flexibility. And while we were collecting these data, this was with a field course in Costa Rica, we had more venom-related experiences, and this involves, rather than snakes, the infamous bullet ant, Paraponera clavata. This is one of the largest bodied ants in the world. And we were working in the arboretum of the La Selva Biological Station, and that's shown here in this photograph. This is a great site for us because it had lots of thief ant colonies. Unfortunately, it was also home to lots of bullet ant colonies. And bullet ants like to nest at the base of trees, and their typical behavior is for a forager to leave the nest and climb up the tree trunk into the canopy. We were out collecting data at night, and what we learned the hard way was that these ants are perfectly happy to let themselves drop from the colony onto the necks of unsuspecting researchers. And so we had to stall our data collection several times to carefully flick bullet ants off of people's necks before they could get stung. It's a very, very painful stinging ant. Okay, so let's move on to the third chapter, and this is going to be an exploration of individual diversity, and we're going to be looking at the effects of body size in ants. And what we observe is that ant species differ in their average body size, and within some ant species, workers further vary in body size, and even nestmates can be strikingly different in size. So we were interested in testing whether body size predicts thermal tolerance. This could be important because for within colony variation, colonies may be forced to adjust their activities to accommodate the different temperature sensitivities among their own workers. And body size differences across species might influence how they interact with each other, and changing climates might differentially affect species and change their species interactions. So we did this work on 2 species of seed harvesting ants in the genus Messor. We did the work in the Negev desert of Israel, a very extreme temperature environment and one that's subject to climate change. And the 2 ants that we worked on share and compete for a single resource, which are the seeds of desert plants. So just looking in this photograph at one of the species, we can see that there is very wide variation in body size within colonies. So this is the smaller bodied species. We're looking at a group of workers at the nest entrance, and you can see that there are relatively small-bodied and large-bodied workers that are quite distinct from each other in size, and the 2 species also differ from each other in size. So we measured the CT max and CT min of workers from different body sizes, and what we found was that, as we expected, the species differ in average body size, although there is some overlap. And importantly, body size effects on thermal tolerance, and that's indicated by the red lines here, those effects were significant for the smaller bodied species, but not for the larger bodied species. And what this suggests is that rising temperatures will be challenging to the smallest workers, particularly the smallest bodied workers of the smaller species. And because the species differ in how worker body size relates to thermal tolerance, rising temperatures could shift the competitive relationships between these species, potentially by eliminating the activity of the smallest bodied workers of the smaller species. And again, continuing on the theme of venom, we had plenty of adventures when we were collecting data at night out in the desert in the Negev. We encountered these very large-bodied trapdoor spiders. These made us a little bit nervous, but it wasn't until we brought out a UV flashlight that we realized that the desert was absolutely crawling with scorpions. Luckily, scorpions glow, as you can see in this photo here, under UV light, and we realized that they were very abundant, so we always had to have a UV flashlight carrying watcher on hand to make sure that the scorpions were staying away from us when we were collecting the data. Okay, so I'd like to just recap a little bit and talk about some take-home messages from the talk. One of the main things that we've learned is that species' experiences with their thermal environments seem to have shaped their ability to cope with extreme temperatures. So some species, like the army ants we looked at, show suggestions of fixed evolved responses to temperatures, whereas other species, like the thief ants, showed evidence for individual flexibility. And finally, the desert ants showed us that changing climates are likely to differentially affect species and may end up causing complex shifts in species relationships and ecology. So I'd like to thank you for joining me on this ant odyssey, and may you avoid venom.